Hello, this is Yashveen. In this video, I'll be solving paper 4 variant 1, May-June 2021, Cambridge IGCSE Biology. 1a. Baker's yeast Saccharomyces cerevisiae is a single-celled organism that is classified in the kingdom fungi, and figure 1.1 is a diagram of a section through a yeast cell. State one other kingdom that contains organisms that all have the same structures. A kingdom that also has cell wall, as you can see over here. Hydrite plant cells, as they also have a cell wall, mitochondria, and other organelles which are present in here. 2. Table 1.1 shows some of the cell functions. Complete table 1.1 by naming the cell structure responsible for each of the function and give the letter that identifies each cell structure. So storage of gene is in the nucleus in the form of DNA. And aerobic respiration occurs in the mitochondria, and amino acids are assembled to make proteins in the ribosomes. The relative labelings of the organelles over here. B. A student made a drawing of one E. coli bacterium. Figure 1.2 shows the student's drawing. The actual length of the bacterial cell is 2 micrometers. Convert the actual length of the cell to millimeters. So you've got 2 micrometers, which is equal to 2 times 10 to the power of -6 meters, and you multiply that by 10 to the power of 3, which gives you 2 times 10 to the power of -3 millimeters, and that is 0.002 millimeters. To state the other information which the student needs in order to calculate the magnification of the drawing in figure 1.2. So magnification is equal to the image size or length of the image divided by the actual length, or the length of the bacterial cell in this case. So you need the length of image. C. Describe the similarities and differences between the structure of the yeast cell and the structure of the bacterial cell. Use information in figure 1.1 and 1.2 in your answer. So right off the top of your head, you can write that their similarities include that both of them have cytoplasm, ribosomes, and cell wall. Since the question is of 6 marks, I'd like to write 3 similarities and 3 differences. And as for the differences, the yeast cell has DNA in the nucleus, whereas bacterial cell does not have a nucleus. The DNA lies free in the cytoplasm. Yeast cell will have linear DNA like all eukaryotic organisms. Remember that bacteria are prokaryotic organisms and yeast cell would be a eukaryotic cell, so it has linear DNA, whereas bacterial cells have circular DNA. Bacterial cells, as you can see, do not have any membrane-bound organelles like the mitochondria or vacuoles or anything like that, whereas the yeast cell does have them. So yeast cells also have membrane-bound organelles, which the bacterial cell does not. I'd suggest that you draw 2 columns for the differences, like I have separated them. Some bacteria are involved in the nitrogen cycle. Figure 1.3 shows part of the nitrogen cycle. State the processes that are represented by 1, 2, and 3 on figure 1.3. So process 1 converts nitrogen from the air in the form of gas into useful ammonium ions. So that is nitrogen fixation. And 2 is going to convert amino acids into ammonium ions by removing ammonia, and that is deamination. And 3 is converting ammonium ions to nitrate ions. This is called nitrification. 2 large quantities of plastic waste are polluting the oceans. A survey published in March 2018 showed the increase in public waste in Pacific Ocean. One area of the Pacific Ocean is known as the Great Pacific Garbage Patch. Data were collected from areas inside and outside of GP, which is short for Great Pacific Garbage Patch, between 1965 and 2015 to estimate the quantity of plastic waste, and the results are shown in figure 2.1. Describe the results of the survey in figure 2.1. So in this question, you are supposed to, since it's asking you to describe, you can describe the trends, which you can see in the question. So first off, we can tell that the amount of garbage inside GF GP, represented by the black bars, are always greater than the amount of waste outside of the GP, represented by the gray bars. And the waste inside of the GP also increases after 1975 and remains constant in these 2 years. So those are the trends. And also, as you can see, the waste inside GP, represented by gray bars, increases and then decreases and then increases. So you can write about how it's going to fluctuate throughout the years. So here's my answer. I've mentioned how the waste will increase from 1985 up until 2015 from around 0.4 kg per kilometer square to 1.2 kg per kilometer square, just to mention some of the data that's given in the figure 2.1. The green turtle, Caloneum medas, is a species of marine animal that is harmed by plastic waste. Figure 2.2 shows a green turtle swimming past a plastic bag in the Pacific Ocean. 1. Turtles are classified as reptiles. State one feature shown by all reptiles that is not found in amphibians. So the reptiles have scaly skin, rubbery eggs, internal fertilization, and they also lay eggs on the land, whereas reptiles do not. You can write any one of them. To outline the dangers of the non-biodegradable plastic waste to the marine animals such as green turtles. So the non-biodegradable plastic does not break down. It is going to remain intact in the ocean, and it could get wound up around the gills or mouth of the animals in the ocean or the marine animals. This will make it difficult for the animals to breathe and gain nutrients through their mouth. If the non-biodegradable plastic is ingested, it can be poisonous, it can be toxic to the health of the marine animals, and it can also be, since it cannot be digested by these animals, it can accumulate in the food chain as other animals eat the animals that have consumed the non-biodegradable plastic. 3. Suggest ways to reduce the quantity of plastic waste. So this can be done by using biodegradable plastic, so it can be broken down and there won't be any plastic waste, and recycle waste. Three, bacteria are used in many biotechnological processes. A, explain why bacteria are useful in biotechnology. So biotechnology is going to use bacteria by, for example, taking the gene for insulin from human cells and then inserting those into the bacterial cells, so that these bacterial cells would be able to replicate when placed in fermenters and be able to produce insulin. And it's usually done that way because there are no ethical concerns that are raised by doing this process, and the bacteria can easily replicate. So as they replicate, they'll be able to produce more of the desired product, like insulin in the example that I've mentioned over here. And they also have plasmids, and you need to use these plasmids to be able to carry the gene for example, the human insulin from the human cells into the bacterial cells. B. Insulin is one of many human proteins that are made by genetically engineered bacteria. Some people cannot produce insulin because their immune system has destroyed the cells that make insulin. 1. State the organ that contains the cells that have been destroyed, that is the pancreas. 2. State the name of the disease caused by the destruction of these cells, which is type 1 diabetes. I suggest that you do write type 1 diabetes and not just diabetes, because this is an autoimmune disease, whereas type 2 diabetes is not. 3. State the function of insulin in the body. So insulin is responsible for reducing the blood glucose concentration following for example, a meal which was high in glucose. the glucose concentration of the blood will increase, and insulin will convert this excess glucose in the blood into glycogen stores into the cell, and as a result it will reduce the blood glucose concentration. See, genetically engineered bacteria that are used to make insulin were grown in a fermenter for 5 days. Samples are taken from the fermenter every 6 hours, and the number of bacteria in 1 millimeter cube of the nutrient solution were counted. Changes in the numbers of living bacteria in the samples taken from the fermenter are shown in figure 3.1. One complete figure 3.1 by adding labels for the axis 1 and 2. The independent variables, so the time, and I've used days in this case, and on the y-axis you've got the dependent variable or the number of bacteria, and as mentioned here in the question, you're counting the number of bacteria in 1 millimeter cube, so I've got per millimeter cube over here. To state the names of the stages of the population growth of the bacteria labeled P to S. So the stage P is the lag phase. Here the population grows very slowly because there are only few cells of the bacteria to grow. And Q is the log or exponential stage where the rate of reproduction is high because there are no limiting factors and there's no competition. There's plenty of nutrients for all of the bacteria to grow. In stage R, the rate of birth is equal to the rate of death, so this is the stationary phase, and the number of bacteria will remain the same. In stage S, the number of bacteria will decrease, and this is the death phase where the birth rate is less than the death rate. With reference to figure 3.1, why the bacteria did not grow in the fermenter for longer than 5 days. So bacteria do not grow for a very long time in the fermenter because in this case, for example, when you have got the stationary phase and the death phase, the birth rate is at least not greater than the death rate. In the stationary phase, the birth rate is equal to the death rate, and in death phase, the birth rate is less than the death rate. And that's because growth of the bacteria in this fermenter is controlled by limiting factors like nutrients, oxygen, and the space to grow. Bacteria do not grow for a very long period of time due to competition with other bacterial cells. So this could be because of lack of nutrients and lack of oxygen, which they do need to grow. And also because as the bacteria grow, they will produce toxic waste substances which build up. The mineral salts are important in the human diet. One of the most important is iron. Explain the importance of iron in the human diet and the effects of iron deficiency. So you need iron to make hemoglobin for red blood cells, which you need to carry oxygen or transport oxygen throughout the body. And as a result of iron deficiency, a person can suffer from anemia. Figure 3.2 shows a field of cassava, manihot esculenta, which is a crop plant grown in parts of Africa and Asia. The plants store starch in their roots, which form a large part of the diet for many people. Cassava does not provide any vitamins or mineral ions. Genetic engineers have modified cassava to increase its iron content. They have done this by incorporating a gene for a membrane protein from the plant Arabidopsis thaliana. One, state the name of the enzyme that is used to cut out the gene from the DNA of A. thaliana. So restriction enzyme is the enzyme which cuts strands of the DNA. 2. Describe how the gene from A. thaliana and the DNA from cassava form the recombinant DNA. So the restriction enzyme which cuts the gene out of the A. thaliana will be used to cut the DNA of cassava plant so that sticky ends are produced in both of the DNA, and this will produce sticky ends that are complementary to each other so they can bind to each other, and DNA ligase will then insert the gene from A. thaliana to the cassava DNA. 3. Scientists who develop genetically engineered varieties of crop plants often breed them for several generations before releasing them for farmers to use. Suggest why the scientists do this. So this will ensure that the genetically modified plants do not have any adverse effects and that the new trait is inherited by all of the offspring plants that are grown in the several generations, so that farmers will not have to worry about the future offsprings and the future generations not having the trait. Two identical potted plants were used to investigate plant responses. Plant A was placed on a clinostat that continually rotated. Plant B was not rotated. Both plants were then placed on their sides and kept in the dark. Figure 4.1 shows the two plants at the start of the experiment and after 7 days. So at the start you have the same appearance of the plant, whereas after 7 days both of them do have grown to a taller length, but plant B, as you can see, is tilted away from the ground, and that's because the growth of plant can be controlled by light and gravity. In this case, the environment factor of light is not different for both of the plants, whereas by rotating the plant A, you are going to ensure that the effect of gravity is same on all sides of plant A. But since plant B is not rotated and it's placed horizontally, or it's placed on its side, that means oxygen is going to accumulate on the lower side of plant B, and this will cause a greater increase in length of the lower length or the lower side of plant B, and so it will curve upwards. So this is an example of gravitropism, where plant is growing away from gravity. A states the name of the response shown by the shoot of plant B, and this is gravitropism. B. Explain the reason for constantly rotating A, as this has changed the growth of plant A, as the light conditions are the same and they're not different. That means this has to be because of gravity, and this will ensure that gravity has the same effect on all sides of A, so that you can, at the end of the experiment, you can compare the results of plant A to the results of plant B. C1 state the name of the plant hormone that causes the response of the shoot of plant B, that is auxin. To explain how the plant hormone causes the response of plant B. In this question, you're supposed to explain the mechanism, the working mechanism of the hormone auxin, or in other words, how it causes response to conditions like light and gravity. So I'd start by stating how auxin is made by cells in the tips of the shoot, and then it diffuses away from the tip. In this experiment, because of gravity, auxin will collect on the lower side of the stem, and this stimulates cell elongation. As a result, the plant bends upwards or away from the gravity. D. Seeds germinate in the soil. The seedlings that grow from seeds show the same response as shown by B in figure 4.1. So the seedlings show the same response would mean that the seedlings also grow away from gravity. Explain the advantages of this response to the survival of seedlings and mature plants. So in other words, explain the advantages to the seedlings of growing away from the gravity. So as the seedlings will grow away from the gravity, they will be able to access more of the sunlight and carbon dioxide present in their environment for photosynthesis. They will also be able to access more oxygen for respiration. 5a figure 5.1 shows the female reproductive system. Label figure 5.1 using the letters listed to show the position of the organs that are identified by their functions. The first one has been completed for you. So Q, the site of fertilization, is the oviduct, because as the eggs are released from the ovary, they will travel away from the ovary and in the oviduct, and this is where the sperms will meet the eggs. And R, the site of implantation, is the endometrium. This over here can be R, the right or the left hand side, the darkest area. And S is the site of estrogen secretion. Estrogen is secreted from the ovaries, so either one of them. And T is the site where sperm are deposited during sexual intercourse, so that would be the vagina over here. B figure 5.2 shows a section through an egg cell at the time of ovulation. 1. Explain why the egg cell contains stores of protein and fat. So this fat will be used by the cell after fertilization to provide energy for mitosis, so that the cell will be able to divide and to make new cells, and protein will be needed inside of the cell or by the cell to form intracellular function like form membrane proteins and make enzymes. To describe the function of the jelly coat, so this jelly coat will harden around the egg cell after fertilization, and it will prevent any more sperm entering the egg cell. C. Fertilization results in the formation of a zygote. Describe how an embryo is formed by a zygote. So embryo is a ball of genetically identical cells, whereas zygote is the cell which forms after fertilization of the egg cell and the sperm cell. So once the egg cell and the sperm cell have fused together to form the zygote, the DNA of the zygote will replicate, and this will be followed by mitosis. Mitosis again is the nuclear division, so this will divide the two sets of DNA into two, and this will form two identical sets of DNA. Then the zygote cell will split into two cells, and then this division is going to be repeated. Eventually, a cluster of genetically identical cells will form, and that is the embryo. Antibodies are proteins that are produced by lymphocytes. Antitoxins are antibodies which neutralize the toxins released by some bacteria. So some bacteria will enter the body, produce toxins, and lymphocytes will produce antitoxins which will neutralize the toxins. The transmissible disease diphtheria is caused by a bacterium that releases a toxin that can cause serious damage to the body. A person is suspected of having caught diphtheria. At a clinic, the person is given an injection of antitoxin antibodies that provide protection against the diphtheria toxin. She is also given an injection of the vaccine for diphtheria. A few weeks later, she is given a second injection of the diphtheria vaccine. Figure 6.1 shows the changes in concentration of the antitoxin antibodies and the antibodies produced in response to the vaccine. Alright, this vaccine will contain dead or harmless pathogens, which are bacteria in this case, that will secrete the toxin. The antitoxin antibodies over here will only neutralize the toxin, which means that any bacteria which are present in the body of the infected person can make more of the toxin. Whereas vaccine will contain again dead or harmless pathogens, it will cause an immune response which will kill the bacteria. So as the bacteria will be killed, no more toxin will be produced, and the antitoxin antibodies will not be needed anymore. So the injection of antitoxin antibodies is given just at the beginning and not later on, and you're supposed to explain the advantage of giving the person an injection of antitoxin antibodies. So this goes back to the point of vaccine. The vaccine is given to the infected person so that it will cause an immune response which will produce antibodies and memory cells, but this is going to take some time, and during this time to kill the antitoxins that have already been produced in the body of the infected person, the antitoxin antibodies are given so that this will neutralize all of the toxins that have already been produced. To explain how the two injections of the vaccine result in better protection against diphtheria than the injection of antitoxin antibodies. Again, antitoxin antibodies will only neutralize the toxin, whereas the vaccine will kill the bacteria, and this will also produce memory cells which will provide long-term immunity or active immunity. And this means that if the same bacteria enter the body again, the second time, memory cells will be present in the body to initiate an immune response which will kill them. So the person will not catch the disease a second time if they have received the vaccine. The answer to A part 2 is really essentially just comparison between active immunity and passive immunity, and you're supposed to describe how active immunity is more beneficial. B. Explain how antibodies protect the body against pathogens. Antibodies are the defense against pathogens that are specific to the antigens that are present on the pathogens. So antibodies can bind to these antigens that are complementary to the shape of the antibodies. This will attract phagocytes which will kill the pathogen and protect the body of the person. C. Antibodies can travel through the body in lymphatic system. State 2 functions of the lymphatic system other than defense against diseases. So other than transporting antibodies, the lymphatic system also transports fatty acids and glycerol that are produced following the digestion of lipids, and after the fatty acids and glycerols are absorbed in the lacteals in the gut. And lymphatic system also returns lymph into the blood. And this is it. We are done with this paper. Thank you for watching.